Good evening, folks. I hope this finds everybody well. This is going to be the first lesson in our section on heredity and genetics. If you are taking my General Biology 1 class, I strongly recommend that you go on Blackboard and get a copy of the handout that goes with this material so that you can follow along with important concepts and some diagrams throughout. Actually, there are two handouts that go with this material. If you're in my class, there is one on Mendelian inheritance, which has basic concepts and vocabulary, and there's a separate handout on Punnett squares, which will show some visuals of the sort of genetic charts that we are going to be making. Also, I would recommend, if you have not already listened to my preceding lectures on DNA, RNA, and cell division, if you haven't gotten any of those at some point, please make sure you go and get them. I'm assuming you understand concepts related to those things for the purpose of this discussion. All right, so we've been talking a lot about the various ways in which our bodies store and move around information. We discussed the DNA code, how DNA chemically stores information on how your body needs to go about carrying out its processes and building its structures by containing the instructions for all of our polypeptides. We have seen through how the process of gene expression, utilizing RNA as an intermediary molecule, we can convert that information from just instructions coded in DNA to a form that we can use to decode and build functional protein molecules that can then do things in your body. We've even talked about mutation as the origin of novel genes. Let's think about that last part for a moment. You do not look like the person who is sitting next to you. If you're sitting next to another person at the time, I'm next to a cat, but you know, I don't look like her either. But in any case, every human is different. Now, we are more alike than we are different, genetically speaking. You probably all heard it said that we as humans share 99% of our DNA with chimpanzees. That is absolutely true. You share 99.9% of your DNA with the other people around you. But for those portions of genes that are uniquely human, like the 1% of the genome that is a human set of genes, there's a considerable range of variation. Now, even in that, some of us, we have more things in common than not. There are certain traits of you that are always the same. All of us have a four-chambered heart. All of us have two eyes. These are things that do not vary from one member of Homo sapiens to another. For about 64% of our uniquely human genes, there are things that just straight up, there is no room for variation. It is the same for every individual. Now, we are interested, of course, in the small percentage of our genes, about 36% of the uniquely human genes that we talked about there, that are, in fact, variable from one person to another. Mutation is going to be a thing that introduces variation, as you've seen. Mutational events can cause new genes to appear, whether those genes are better genes, worse genes, or selectively neutral genes. But once a new gene appears via mutation, it's now part of the population. It's there, and as long as the individual who has that new gene can reproduce, then that gene can be passed on to others. It's now an option circulating in the world around you. And what this means fundamentally is that there's more than one option for how to do a given thing biologically speaking. The reason you look different than the person sitting next to you is because you have slight variations in a lot of those genes that make little details of your body, like eye color and hair color and height and so forth, different from one another. That is getting us into the topic of this discussion, which is the topic of heredity and inheritance. Heredity is the transmission of genetic information from one cell to another. Most often that means genetic inheritance, that is passing things from parent to offspring. Humans cannot engage in horizontal gene transfer like bacteria can. We do not have plasmids that we can pass from one individual of our species to another. What we can do, however, is that you can combine your DNA with somebody else's DNA during sexual reproduction and produce offspring that have a unique combination of half of the genes from one parent and half the genes from the other. But that brings us to some really important questions. Why is it that you have the features that you do? Why is it that some features are easier to pass on than others? These are the things we're going to be trying to address in the course of this chapter. All right, now let's think about the basics here. For a long time, we didn't really have any good understanding of how heredity worked. Consider that we didn't even know DNA was a molecule until the 1860s. We didn't even realize that it was coding for genetic information until the 1950s. So we didn't really have a good sense of the mechanics of all this for much of the early history of the biological sciences. As recently as the 19th century, there was a common idea in biology called the blending hypothesis. And the idea behind the blending hypothesis is that you were kind of a genetic average of your parents, that the features of mom and dad sort of just averaged out in you to make the features that you have. Now that sounds okay at first glance. You might think, okay, yeah, I've got mom's nose and dad's eyes. It sounds like I'm kind of a blend of their traits. But blending doesn't account for a lot of things. If you're just averaging out the features, why is it you have dad's nose and not a nose that's somewhere between yours and dad's? How is it that two blue-eyed parents can have a brown-eyed child? Actually, that can't happen. Let's go the other way. Two brown-eyed parents can have a blue-eyed child. For that matter, why can't two blue-eyed parents have a brown-eyed child? Why is it that your grandma might have blue eyes, your mom doesn't, but then you do? How is it that two short parents can have a tall kid? None of these things can be accounted for by genetic blending. There's clearly something else going on. And as early as the 1700s, the nascent biological sciences were realizing there was something we didn't know, that there were factors involved that we simply did not understand, and we needed to figure out a mechanism for genetic inheritance. And a number of biologists tried to do this. In the early 1800s, one of the great names of early biology was a French scientist named Jean-Baptiste Lamarck. That's Jean dash Baptiste, Baptiste, Lamarck, Lamarck. Jean-Baptiste Lamarck. He was a French scientist employed at the French Museum of Natural History. He was an expert in invertebrate paleontology. He's considered one of the fathers of the science of paleontology, the study of fossil organisms. He was a very important early figure in cell theory, conceiving a lot of ideas essential to that. And he is also remembered as the guy who came up with the term biology for this thing that we are doing. Now, Lamarck published a book in 1809 in which he endeavored to solve the problems of both evolution and heredity. How did inheritance work, and how did species change over time? Lamarck was an evolutionary theorist in the vein of Darwin. Darwin actually believed a lot of Lamarck's ideas early on until his own research demonstrated that they didn't work. And Lamarck's book on the topic was actually published the year Darwin was born, 1809. But Lamarck's theory of heredity and evolution was kind of a unified thing and involved two basic components. First off was the idea of the inheritance of acquired traits. Careful of the spelling of the word acquired. A-C-Q-U-I-R-E-D. Inheritance of acquired traits. And what this meant was that Lamarck believed that anything that happened to you during your lifetime, that changed your body in some fashion, could then be transmitted to your offspring. That changes to your morphology could be carried on to your kids. So he said a guy who works out hard in the fields and gets big and strong and healthy will have stronger kids. He pointed to an example from his own life. Lamarck was a soldier before he was a scientist, retired from military service due to an injury in one leg. But then it turned out he later fathered a son who had a deformity in the same leg. And so he said, well, clearly, look, evidence that my deformity was passed on to my son. The other principle of Lamarckian inheritance, or Lamarckian genetics, as you might think of it, is going to be an idea called use and disuse. And the idea behind this is that if an organism makes frequent use of a part of its body or a feature of its body, then that part or feature will be enhanced in subsequent generations. It will become better over time. If, on the other hand, the organism does not make frequent use of that part of its body or that feature, it will atrophy and it will become less prominent over time and might even eventually disappear. By way of pointing out the argument of use, he pointed to the giraffe, suggesting that the ancestor of the giraffe had been an animal with a much shorter neck that stretched its neck trying to get to higher and higher leaves in the trees. And as a result of that effort, its descendants were born with a longer neck to meet that need. Likewise, he looked at the human tailbone and said, as a vestigial structure, clearly this was evidence that we had had a tail at some point in our ancestral past that was no longer necessary, and therefore it began to decline, leaving behind only the stubby little tailbone that we have today. Now, you already know, if you've been paying attention to the things I've been telling you about, that Lamarck was wrong. There's nothing about stretching my neck to try to get to higher leaves that is going to modify my DNA in such a way that my offspring will be born with a longer neck. Likewise, making frequent use of a part of my body is not going to cause the genetic code for it to be rewritten to make it better or stronger or larger. And likewise, just because I don't make use of a part of my body does not mean that that part of my body is going to just decline over time. As you have seen, our DNA is remarkably good at preserving even defunct genes and carrying them on. So Lamarckian evolution was incorrect. It was actually demonstrated to be incorrect by a famous German biologist by the name of August Weismann, W-E-I-S-M-A-N-N. Weismann did a famous experiment in which he cut the tails off several generations of mice in an effort to see if he could eventually breed tailless mice, and found that in fact he could not do so. But Lamarck had been right about one very big thing. Namely, he had made an important link between adaptation to the environment and the features that are successful. Charles Darwin would later build on that idea to develop his concept of natural selection. In many ways, Lamarck kind of put the cart before the horse. He said environment causes a threat, and your DNA responds, or your genetics respond, he didn't know about DNA, by creating new features to meet that need. Rather, Darwin realized it's kind of the other way around, that mutation produces random new features, and then the environment filters which ones are useful and which ones are not. That's what we call natural selection. Nature is selecting the useful traits. So although Lamarck was wrong about his theory of inheritance and evolution, he provided important inspiration to later scientists who then built on his ideas, and he also realized that very all-important connection between adaptation and features that succeed or fail. Now, another guy who's going to make a stab at this is going to be Charles Darwin himself. We're not going to go into Darwin's evolutionary theories in here. We touched on them a little bit in my chapter on mutation and cancer, if you haven't heard that already. And if you take my subsequent course, Biology 2, we talk about evolution a great deal in there. But we also should know about this, that in addition to his idea of natural selection, the very famous idea that Darwin is famous for, that he also attempted to solve the problem of inheritance, to figure out how genetic inheritance worked, how heredity worked, how features were passed from parent to offspring. Darwin came up with a hereditary theory called pangenesis, P-A-N-G-E-N-E-S-I-S. And the idea behind pangenesis is that Darwin thought that every part of your body produced little particles that he called gemmules, G-E-M-M-U-L-E-S, and that those gemmules would then be collected in the gonads, the reproductive organisms, in order to teach that organism how to make the parts that it needed. That, present in the male and female reproductive cells were all of the gemmules that would then be transmitted to the offspring, so that the offspring knew from those discrete particles produced by every part of your body how to build that part of your body. Darwin's theory of pangenesis also did not work. It was actually disproven by his cousin, Francis Galton, G-A-L-T-O-N, who did an experiment in which he took black rabbits and took samples of their blood and injected it into white rabbits, figuring that the gemmules for black fur would then be transmitted to the white rabbits, and they would have black offspring, which they did not. So Darwin actually went to his grave never knowing how inheritance worked. But the guy who solved the puzzle of inheritance lived at the same time as Charles Darwin, and he was a relatively humble monk, a Catholic priest living in what is today Austria, by the name of Gregor Mendel, G-R-E-G-O-R, Mendel, W-E-N-D-E-L. Gregor Mendel was born in what is today the Czech Republic, but spent most of his life in Austria. He was born under the name Johann Mendel, but his family was unable to afford to send him to college, and so in order to get an education, he joined an Augustinian monastery, the Augustinian order of the Catholic Church, then as now highly prized education, and they would pay for his education if he became a monk. So Mendel enrolled in the order. He took on the monastic name Gregor rather than his birth name of Johann, and spent his life as a Catholic priest and a monk at the monastery. The monastery sent him to the University of Vienna, where he studied statistics and physics under Johann Christian Doppler, who was the guy who discovered the Doppler effect, like radar. But anyhow, though, Mendel returns home to his monastery. He lives a quiet life. He does some school teaching around the local village, but he actually never passed his teaching certification. He could not become licensed as a teacher because he had severe anxiety and failed the test due to test anxiety every time the examination came up. But at the monastery, Mendel's duties included tending the gardens. His family had been horticulturalists, and in fact, he had briefly considered following his father's career as a gardener. So he was good at this kind of thing, but he's looking at the plants, and he's getting curious. And he's saying to himself, how do features get passed from parents to offspring among the plants? What determines which flower color will be present in any given new flower? What determines the length of the stalk or the shape of the leaves or whatever else? And Mendel did some reading, and he learned that the problem of inheritance had not been well solved, that the blending hypothesis clearly had been shown to have problems, but nobody had yet come up with a better alternative. Now, Mendel decided to set up a series of experiments to try this out for himself. And between the 1850s and early 1860s, Mendel spent 7 years experimenting with plant breeding. He chose for his primary subject the common garden pea plants of the genus Pisum, P-I-S-U-M. He chose the garden pea for a couple of reasons. One, because they grew fast. He could produce several generations every year in the greenhouse of the monastery, which helped him to get quicker results. Two, because the garden pea had an easily removable stamen, S-T-A-M-E-N, if you don't know what that is. The stamen is the male reproductive organ of the flower. Mendel could take the stamens out of the flowers of one plant and deliberately control which other plants it's pollinated, so he could keep track of which plants were breeding with which other ones. That was useful for his experiments, necessary for his experiment. And finally, and perhaps most importantly, common pea plants have a lot of either or characteristics, things that vary just, like a nice, neat, you have this one or that one with no middle ground in between. So, for example, they have either yellow-skinned peas or green-skinned peas, or pea plants with white flowers or pea plants with purple flowers, no intermediate colors. Mendel identified about 7 different traits of his pea plants that were either or variation. He knew very well that what he was going to have to do in order to set up his experiment was to have clear, unambiguous results. And so he knew if it was a trait that had like a lot of middle ground where it's like, okay, is this more purple or more white? It would be very hard to interpret and figure out the inheritance pattern. So he focused on his research with traits that had clear, distinct variation. Mendel set up a very clever experiment. It was very, very well designed. And before I tell you about his experiment, let me give you some terminology that we're going to be using throughout this section. Some terms that I'm going to use periodically. First off, let's talk about a thing called a character. C-H-A-R-A-C-T-E-R. A character is any heritable feature. It is any feature of your body that is genetically encoded and can be inherited by your offspring. So eye color is a character. Number of fingers on each hand is a character. Skin color is a character. All of these are heritable features. They're things that you get from your parents and are passed on to your offspring in turn. Things that are subsequent modifications of your body are not characters. If I have a cool cyborg eye that I got after I got into a fight with a robot and now I've got an eye that can shoot lasers and stuff, that's not a character. If I have a tattoo, that's not a character. If I have a scar, that's not a character. Those are all things that are not heritable features of my body. Even some of the things you're born with are not actually heritable features. Fingerprints, for example. Fingerprints are not genetically encoded. They're a result of fluid forces acting on your developing skin in utero, and they are not a feature of your genetic inheritance. Everybody has them, but the specific pattern you have is not encoded in your DNA. Now, for about 64% of human characters, as I indicated a little bit earlier, there is no variation. That you and everybody else in our species has exactly the same feature for that particular character. Like, for example, we all have 4-chambered hearts. We all have paired kidneys. We all have 32 teeth in the adult mouth. These are standard features of our species. These are the things that do not vary from one individual to another. Those things are not particularly interesting for genetic studies because every single human has the same genes for them, barring, novel mutations. We're interested in the 36% or so of our DNA that is variable from one individual to another. And that was what we're going to be focusing on. And that means that basically, if there's variation involved, there can be more than one option for a given character. That's where we get into the term trait, T-R-A-I-T. A trait is a specific form of a character. So, for the character of eye color, I might have the trait of brown eyes or blue eyes or green eyes. These are traits for that character. There's no upward limit on how many traits could exist for a given character. We, of course, are interested in the portion of our genome for which there's more than one trait. If we all have the same trait for the character, then it's not interesting because we're all inheriting the exact same thing from one another. We're interested in those genes for which there is more than one trait available, more than one form of the gene. Now, speaking of, the word allele, A-L-L-E-L-E, is often used synonymously with trait. The allele is the gene responsible for a given trait. So I could say here is a blonde hair allele. That is the specific gene, the specific genetic sequence in DNA that, if decoded and translated in my body, will cause my hair to be blonde. So I could say I have a blonde hair allele, which means that I have the gene which codes for the trait of blonde hair for the character of hair color. Hair color is the category, you might say. That's the character. Blonde hair is the trait. That's the specific form that it can take. And the allele is the gene that makes that happen. Now, it is possible, you might have noticed I pointed at myself and said blonde hair. Doesn't look terribly blonde, does it? Nope. The reason for that is because I am a carrier of the gene for blonde hair. My mom was blonde, my dad was not. If you are a carrier, C-A-R-R-I-E-R, that means that you possess a gene that is not being expressed, that there is a gene located in your DNA that is not being expressed. Some other gene is being expressed instead. I am clearly expressing the gene for the trait of brown hair, but I am a carrier of a gene for blonde hair that I inherited from my mother. What it means to be a carrier, essentially, is that although you personally do not have that feature, your offspring potentially can. You can potentially pass that gene on to them. Now, any given gene is always found at the same spot in the DNA. We call that spot its locus, L-O-C-U-S. If you're talking about the plural form, that is loci, L-O-C-I, sometimes pronounced loci, L-O-C-I. But a locus is the spot in the DNA where a given gene is located. The locus for a given gene is always the same for a given member of our species. So my gene for eye color is in the same spot on the same chromosome as your gene for eye color. That feature, that locus, that location for that bit of information is a universal feature of our species. Even if you and I have different alleles at that locus, we still have an eye color gene of some kind located in that spot. And lastly, a term I'm going to use periodically is the term segregation. S-E-G-R-E-G-A-T-I-O-N. In other words, segregation means something in genetics that it's different than what it means in day-to-day life. In genetics, the word segregation refers to a situation in which different offspring of the same parents possess different traits. So my dad had brown hair and my mom had blonde hair. I have brown hair. My sister came out blonde. So that is clearly an example of segregation happening amongst us. All right, so this is some basic terminology that I wanted you to know before we get started with anything else, before I tell you about Mendel's experiments. We will be introducing other terms as we go, but I thought that was some good stuff to have up front. All right, now let's talk about how Mendel did his research, and then we'll talk about what he learned from it. Mendel, again, knew he wanted to try out seeing the inheritance of unambiguous traits, things that it was clear, okay, you're either purple flowers or white flowers with no intermediate color, green peas or yellow peas with no intermediate color. So Mendel identified all the traits that he wanted to study, kind of paired them up with each other. He then found a plant that had each trait he desired. He chose to focus on only one trait at a time because he realized, or one character at a time, I should say, not traits. He said focused on one character at a time because that way he was not muddying the waters by saying, okay, well, is something different happening with the flower color at the same time that something's happening with the pea color? Now, he did do another experiment later that involved that, but we'll talk about that further down the line. But at his initial exercise, he looked at only one character at a time. He'd say, okay, I'm setting up an experiment for flower color. And he wouldn't worry about color of the peas or texture of the pea pods or anything else while he was doing that. He would only concern himself with the color of the flowers. He would then pick a representative plant for each color that he wanted to study and say, all right, here's a white-flowered pea plant and a purple-flowered pea plant. Those are the two variant traits that pea plants have. And he would take the plant that was the representative example of each one, and he would allow it to self-pollinate, plant the seeds, and then kick out any offspring that came out with the other traits. So he would allow a purple-flowered pea plant, for example, to self-pollinate, and he would throw out any offspring that came out white rather than purple. And he did this for several consecutive generations until he was only getting the trait that he wanted. So that is, he had a white-flowered pea plant that always produced more white-flowered pea plants when it self-pollinated, and he had a purple-flowered pea plant that always made more purple-flowered pea plants when it self-pollinated. If an organism always passes on a given trait, by the way, the term for that in genetics is we say that it is breeding true. If something breeds true, that means that it always successfully passes on that given trait. And he did this for every specific trait that he wanted to study. So he had an example, white-flowered pea plant that bred true, a purple-flowered pea plant that bred true, and then he had a yellow-skinned pea plant that bred true and a green-skinned pea plant that bred true, and so forth. And he did this for every specific trait in the paired groups that he wanted to look at. Once he had satisfied himself that they were always going to breed true when they self-pollinated, he paired them off with their opposite traits, crossed those two plants, and then kept track of what happened to the offspring for several generations. By the time Mendel was done, he had spent more than 7 years on his experiment, and it involved more than 29,000 individual pea plants. But the end result was that Mendel discovered the basic fundamental principles of the science of heredity, of the basic ideas of how inheritance works, how traits are passed from parent to offspring. Now, there are obviously things we have learned since Mendel's time. There were things he did not know about and could not know about with the technology available to him. But at the same time, the basic principles that Mendel laid down in the 1860s stayed with us. By the way, a little tragedy of Mendel's life. Gregor Mendel never got recognition for his discoveries during his lifetime. He presented his paper on 2 separate occasions to 2 small audiences of scientists, altogether a grand total of maybe 40 people, and most of them came away from his lecture not understanding the things he was talking about. Apparently, it was just a little bit too much to fit into a 1-hour lesson, as you're about to see. And so most of them really didn't do much with his ideas. He published his findings in an article called Experiments in Plant Hybridization, but he placed it in a journal called The Proceedings of the Natural History Society of Brunn, B-R-U-N-N. It was a small scientific publication from the city near the monastery, which had almost no circulation. This is kind of the equivalent of putting your Nobel Prize-winning thesis in the newsletter of your local gardening club. But as a result, hardly anybody who was interested in genetics ever knew Mendel's article had been published. Furthermore, in the very few university archives that got copies of the Proceedings of the Natural History Society of Brunn, Mendel's article, because of its title, was filed under plant hybridization and agriculture. As a result, most people who were interested in genetics never thought to look for it where it was. And as such, the simple fact of the matter is that most biologists who would have benefited from seeing Mendel's ideas were completely ignorant of the fact that they even existed. And in his later life, Mendel got really busy with affairs of the church. He became the abbot, the head of the monastery, and never really got back to doing a whole lot of science after that. And so he died, never having gotten recognition for his blindingly important discoveries. Ironically enough, Charles Darwin owned a copy of Mendel's article. It had been sent to him by a colleague in Germany. And Darwin, who was at this point in his career very famous and got a lot of books and things sent to him by admirers, said, "Oh, that looks nifty," and he stuck it on his bookshelf and never read it. We found the book in Darwin's personal library, still tied shut with the strings from shipping, about 15 years ago. Darwin had literally spent the last decades of his life working at a desk within arm's reach of the book that would have solved the single biggest problem with his own theories, and one that he had tried and failed to solve for himself, the question of how heredity works. The moral of the story, folks, is read your damn textbooks. There is important stuff in there. All right, so given that, what happened in Mendel's experiment? So Mendel sets up his experiment. I'm going to just walk you through Mendel's experiment, and we're going to talk about what kind of outcomes he got. So he decides to try flower color first. And common garden peas have two basic flower varieties. They have purple flowers or they have white flowers. No intermediate color. Mendel had been carefully selectively breeding his plants for several generations till he had a true breeding purple one and a true breeding white one. So the purple one always made purple offspring, white one always made white offspring. So Mendel pairs them off and pollinates them with each other to find out how the offspring come out. Now, by the way, the terminology we use for this a lot in genetics, we're drawing a pedigree, P-E-D-I-G-R-E-E, which is like a chart tracing the flow of genetic information over time or tracing the passage of traits from parent to offspring over time. That's pedigree. And when we're doing a pedigree, the starting organisms, whichever organisms we begin with, our starting point for that discussion, is referred to as the F0 generation, capital F subscript number 0, sometimes called the P1 generation, capital P subscript number 1. And the offspring of this first generation is referred to as the F1 generation, F subscript 1. The offspring of the F1 generation would be called the F2 generation, and their offspring would be the F3 generation, and so on. So Mendel's F0 generation, his starting point, is a true breeding purple flowered pea plant, true breeding white flowered pea plant. So he crosses them to see what happens, and he plants the seeds and counts up the offspring. His statistics background, by the way, was invaluable in keeping track of the numbers for the very complex research he was doing. Now, the blending hypothesis was still around in Mendel's time and suggested that what he should have gotten in this instance was pale purple flowers, a flower that was kind of somewhere in color between purple and white, maybe lavender, something like that. That's not what happened. Every single flower in the F1 generation was purple. The white trait had completely disappeared as far as Mendel could tell. There was not a single white flower among the lot of them, and moreover, the purple ones were just as purple as their purple parent had been. There was no pale purple going on. And Mendel says, that was unexpected. I wonder what happens next. So he took those purple flowered plants and he allowed them to self-pollinate. He allowed them to fertilize with themselves to see what came out. When he did that, he found that in the F2 generation, the majority of the plants also came out purple. About 75% of his F2 plants were purple. But about 1 in 4 of them, about 25%, was white. The white trait had somehow skipped a generation. Now Mendel was really curious about what was going on here, so he allowed his F1 plants to self-pollinate as well. And he allowed each purple plant to self-pollinate and each white plant to self-pollinate. And he tracked them for several subsequent generations. And he found a pattern emerged that all of his white flowered plants, no matter how many generations he let the experiment run, they always bred true. White flowered plants, whenever they self-pollinated, always made more white flowered plants. With the purple ones, remember that Mendel had about 75% of his population was purple, 25% was white. Of that 75 that was purple, 25%, 1 in 4 of the original plants, was a purple plant that bred true, that whenever he let it self-pollinate, it always produced more purple flowered plants. The remaining 50% of his original population was a purple flowered plant that, when it was allowed to self-pollinate in the F3 and subsequent generations, produced more of the same thing you got in the F2. In other words, about 50% of them repeated the F2 results, where they come out about 75% purple and about 25% white. Now, Mendel tried this experiment again with other traits. He tried pairing off green peas and yellow peas. Got similar patterns emerging. He tried wrinkly peas versus smooth peas. He got similar patterns emerging. It seemed to work the same way every time he looked at paired off conditions. Now, first thing we can conclude from Mendel's work, first off, is the blending hypothesis is right out the window. At no point was there blending. We never saw the pale purple flower. Secondly, Mendel concluded there's something going on with purple that makes it more common than white, that whenever we pair off purple plants and white plants, it seems like purple wins, that more of them will come out purple than white. That seems to be an important thing. But even a purple plant with a white parent had some kind of white stuff in it. Something was being passed on from the white parent to a purple flowered offspring that stayed in it and allowed that purple flowered plant to later produce white offspring of its own, allowing the white trait to skip a generation. It was clear to Mendel that something from the white parent was present in the purple offspring, even if they were just as purple as their purple parent had been. So Mendel's kicking all these ideas around, and he's trying out his different experiments, and he ultimately comes to several important conclusions about the way heredity works. 5 basic theoretical principles that Mendel ends up discovering as a consequence of all this. And if you have my handout, you can follow along with these. I've listed them all and explained each conclusion. But let me talk about them right now. First off, first thing Mendel concludes: organisms do not pass on their traits directly. Rather, what they are passing on is discrete information on how to make a trait. It was clear that the organism was not passing its traits on directly, because when you bred a purple plant and a white plant, the whites seemed to disappear. The purple offspring were just as purple as their purple parent had been. So the trait itself was not being directly transmitted. But it was equally clear that some kind of information on how to make a white flower was being passed on to the F1 generation, because when the F1 purple flowers were allowed to self-pollinate, they would occasionally spit out white offspring. So something was coming down from the white flowered plants, and Mendel determined that was instructions. It was information on how to make the white trait, even if that plant was not doing so itself. Mendel did not know what form that information took. In his writings, he referred to it simply as factors. Today, of course, you know Mendel's factors as genes, because you know things about genetics that Gregor Mendel did not. So organisms do not directly pass on their traits. Rather, they pass on information on how to make traits. Second big principle coming out of Mendel's work: each organism has 2 factors, or genes as we would call them, for each character, one from each parent. Now you know why this is. Gregor Mendel did not. You know because you get half of your chromosomes from mom and half of your chromosomes from dad, and your chromosomes are paired. Mendel didn't know about chromosomes. They hadn't been discovered yet. But it was evidently clear to him from his research that each parent was making an equal genetic contribution to the offspring. That something, whatever that information was that was being passed on, was being passed in duplicate. That mom passed on a copy of her information, and dad passed on a copy of his information. So Mendel's work clearly showed that you were getting 2 copies of each gene. He didn't use the term gene, he didn't know about the chromosomes making this happen, but he was able to see the pattern. Third principle coming out of Mendel's work: not all genes in the population for a given character are the same. Therefore, as long as there is variation, you have 2 options. You could potentially inherit 2 copies of the same gene, or you could inherit 2 different genes. So in a population with variation, if there's more than one allele out there, if there's more than one gene out there for eye color, and some people have a blue one and some people have a brown one, you're getting 2 copies of each gene, one from each parent, right? This means that you have 2 basic possibilities anytime you reproduce. One is that the child will end up with 2 copies of the same gene. Maybe a brown-eyed parent gets together with another brown-eyed parent. The kid gets 2 copies of the gene for brown eyes, one from each of them. If that is the case, you are said to be homozygous for that trait. Homozygous. Or to put it another way, you are a homozygote. Homozygote. Now, another option here is that you can get 2 different genes. Maybe you got a brown-eyed gene from dad and a blue-eyed gene from mom. If that is the case, you are said to be heterozygous for that particular trait. Prefix hetero means other. Prefix homo means same. And you already know zygote, is the origin of the cell. So homozygous or heterozygous. Now keep in mind, this is a thing that applies to any one specific character, not all of your characters. I can't look at an organism and say that cow is heterozygous. What is that cow heterozygous for? That cow might be heterozygous for fur color pattern and homozygous for eye color. You know, there are variants that exist. And so when we talk about whether you're homozygous or heterozygous, that is a gene by gene thing. It's locus by locus is more than what I should say. So at any given locus in your DNA, you either have 2 copies of the same gene, in which case you're homozygous for that trait, or you have 2 different genes, in which case you are heterozygous for that trait. All right, so far so good. Okay, this brings us to our next condition here. 4th point, 4th basic principle in Mendelian genetics. Organisms, sorry, let's say organisms, let's start back a second. Genes do not blend or influence each other. Rather, your genes remain separate and distinct, and they segregate randomly and independently when the organism that has them produces gametes. I'll give you that again because that's an important principle. Genes do not blend together, nor do they influence each other. Rather, they segregate randomly and independently whenever the organism that has them produces gametes, and they remain separate and distinct. Let's unpack that. So first off, genes do not blend together. There's no blending. At no point did Mendel see the genes of his parent plants combine to make an intermediate trait. So the 2 genes you got from your 2 parents, regardless of what you're actually visibly manifesting, you still have those 2 genes as 2 separate things. If I got brown-eyed gene from one parent and a blue-eyed gene from another parent, my eyes are brown. Well, they're not, my eyes are blue, but, you know, supposing my eyes were brown, but I still have the blue-eye gene in there. It didn't go away. It didn't get gobbled up by the brown-eye gene. It didn't get destroyed. The brown-eye gene did not shut it down. It did not blend in with the brown-eye gene. Those genes remain separate and distinct in my DNA. And when I make gametes, they segregate randomly and independently. And what I mean by that is randomly, that there's no predicting which gamete will end up with which of my 2 genes, and independently, meaning that what happens to any one gene will happen, has no bearing on what happens to any other gene. Whatever happens to my gene for eye color has no bearing whatsoever on what happens to my gene for hair color or shape of my nose or anything else. Now, you know, again, a thing that Mendel did not. You know why this happens this way, because presumably you have watched my lesson on cell division, and you know about meiosis, and you know about a thing called crossing over. Remember, during prophase 1 of meiosis, your maternal and paternal chromosomes, the maternal one you got from your mom, the paternal one you got from your dad, they pair up, they cross over, and they exchange some of their genes back and forth, which means that some of the genes that started off in your chromosome from dad end up in the chromosome from mom and vice versa. And they get mixed up in that way so that whenever your cell splits up the chromosomes during meiosis to make eggs or sperm, half of your genes end up in any given egg or sperm, but because of crossing over being a random process, there is no predicting which specific gene will end up in any given gamete that you have. That's the segregating randomly parts. Furthermore, they segregate independently. There's nothing about having any one particular gene go one way that says that another gene is going to go that same way. There is one weird exception that Mendel never stumbled upon. It's a thing called linkage, L-I-N-K-A-G-E. Linkage is a phenomenon that happens when 2 particular traits are often inherited together. And it's generally happening because the genes for them are literally right next to each other in the DNA, which means that if one of them crosses over into the other chromosome, the other one usually crosses along with it. And therefore, those 2 traits are usually, but not always, found together. They're not literally linked, but they are commonly transmitted as a unit. My favorite example is, some of you might know this already, if you have a cat with white fur and blue eyes, that cat is almost always deaf, because the gene for pale pigmentation in cats exhibits linkage with the gene for deafness in cats. Now, it is not a perfect correspondence. I had a white-furred, blue-eyed cat growing up that could hear a can opening from anywhere in the house that you happened to be. So clearly her hearing worked just fine. So it's not a perfect correlation, but it's a thing that can happen sometimes. But the general rule, however, is that no gene influences any other gene. The inheritance pattern for one has no bearing on the inheritance pattern for any other. All right, and last but not least, the fifth principle that Mendel discovered from his research, and the one that I think people misunderstand the most. It's his best known principle, but it's the one people get wrong the most. Some genes are dominant and some are recessive. Dominant genes and recessive genes. You all know those terms. Let's talk about what they mean. First off, spell it right. D-O-M-I-N-A-N-T. D-O-M-I-N-A-N-T. Dominant, A-N-T, not dominate, A-T-E. Dominate, A-T-E is a verb, saying I am dominating something. I am dominating this chess match or dominating on the football field or whatever. Dominant, A-N-T, is an adjective, saying that this thing is the dominating force, that it is stronger or whatever, you know. Recessive, spell this right as well, R-E-C-E-S-S-I-V-E. Here's what you need to understand. Dominant gene does not mean good gene. Dominant gene does not mean strong gene. Dominant gene does not mean common gene. Dominant has nothing to do with you, the person carrying it. It's nothing to do with your personality, with your health, or any other factor of you personally. It is a chemical feature of the gene itself. It does not vary from one individual to another. If a gene is dominant in me, it is also dominant in you. If you see a guy and his kids all look like him, you can't say, oh, that guy's got dominant genes, because the features that look like him might very well be recessive features, you know. And again, it's nothing to do with a person. It's a specific individual feature of each particular gene. Likewise, recessive. The word recessive does not mean bad gene. Recessive does not mean weak gene. Recessive does not mean rare gene. And it has nothing to do with you, the individual, having it, with your personality, with your features, or with your health, or any other part of you. If a gene is recessive in me, that gene is also recessive in you. It is a chemical property of the gene. So what does dominant versus recessive actually mean? It means one thing and one thing only. Namely, if you are heterozygous, the dominant gene will be the one that is expressed. You got two genes for each trait. Only one of those two genes is expressed. Basically, when your body is expressing genes, basically the normative thing is that one gene out of each pair gets shut down. Now, here's the thing. If you're homozygous, it doesn't matter. You've got a brown gene or another brown gene. If one of those two gets shut down, you're still going to have brown eyes. If you are heterozygous, it matters which one gets expressed because they're two different genes, right? In that case, that's what dominant versus recessive means. Because if the gene is dominant, the dominant gene will be expressed if it is present. The recessive gene will not. It will be there, it will still be part of your DNA, but you will be a carrier of that gene. Only way you express a recessive gene is if you are homozygous for it. In other words, if you have two copies of the recessive gene, if you did not inherit a dominant gene at all, then there's only one choice. Your cells have to express the recessive. It's the only option they've got. So when one of the two of the pair gets shut down, the other one is expressed. They're both the same gene. It's going to be the recessive trait. Incidentally, expressing a recessive gene does not mean that that gene became dominant. It's still recessive. It has not become dominant just because it's being expressed. It remains recessive, which means if you pass it on to your kids, if they get a dominant gene from your partner, guess what? The dominant gene will be the one expressed. The recessive gene did not turn into a dominant gene just because it was expressed in you. It just meant it didn't have any competition. So, given what I've just told you, let's talk about the difference between a thing called genotype and phenotype. G-E-N-O-T-Y-P-E, genotype. Phenotype, P-H-E-N-O-T-Y-P-E. Your phenotype is whatever feature you visibly express. So my phenotype for brown hair is that my phenotype for hair color is that I have brown hair. My phenotype for eye color is that I have blue eyes. Those are phenotypes, visibly expressed features. Now, I say visible doesn't have to be visible to the naked eye. Like, for example, I have type B blood. That is my phenotype for blood type. I can't look at the blood and say that it's type B, but I can do a test and find that. Now, that is the trait that is obvious. Phenotype does not tell you whether or not you have a recessive gene that you're carrying. All it tells us is what you're expressing. Genotype is telling us what genes you have, whether or not they are being expressed. And for any given locus in your DNA, any given spot where a particular character's gene is found, you have three possible options. For any given locus, you could be homozygous dominant, meaning that you have two copies of the dominant gene. If that is the case, you will definitely express the dominant gene. Furthermore, when you pass it on to your kids, so will they, because all your kids are guaranteed to get a copy of the dominant gene from you, and dominant genes are expressed if they are present. That's what it means to be a dominant gene. So, homozygous dominant means you have two copies of the dominant gene. You will always express that trait, so will all of your offspring. You could be homozygous recessive. That means you have two copies of the recessive gene. Because there's no dominant gene to compete with it, the recessive trait will be expressed. You will always pass on a gene for that trait to your kids, which means that they will either be carriers if they get a dominant gene from your partner, or they will express the trait themselves if they also got the same recessive gene from your partner. Basically, homozygous recessive is the only way recessives get expressed. Finally, your third option is heterozygous. You don't have to specify heterozygous dominant or heterozygous recessive, because heterozygous just means two different genes, and you already know the rule for this, that in a case of heterozygosity, the dominant gene is the one that gets expressed. So you could reasonably say all heterozygotes are heterozygous dominant, because that's what it means to be a dominant gene. But we don't bother saying that, we just say heterozygous. And if you are heterozygous, you will express the dominant condition. You will express it just as much as somebody who is homozygous dominant. In other words, there's no difference in the color between a homozygous dominant purple flowered pea plant and a heterozygous purple flowered pea plant. They're both going to be equally purple. The only difference comes in the offspring, in that heterozygotes can potentially pass either gene to any of its offspring, which means that although it may express the dominant gene, its offspring could potentially express the recessive gene. This is how things skip a generation, folks. All right, that is Mendel's basic rules. And we have learned here some really important genetic concepts. We have learned homozygous versus heterozygous. We've learned dominant versus recessive. We've learned what characters and traits and alleles all are. We've learned some basic features about how genetics works. So this gives us a fundamental introduction to this particular branch of genetic science. And I think this is a good time to call this an end point for this lecture. So we will stop here. Next time we will talk about Mendel's experiment again. We'll go back to Mendel's experiment, knowing what we know now. We'll see why he got the results that he got, and we will learn how to predict the results of genetic exercises of our own using a tool called a Punnett square. I strongly recommend do your reading over this. If you're in my class and you have the textbook, look over that handout, those two handouts that I told you about, and make ready for this, because this is going to go a lot better if you're familiar with the basic structure. All right, hope you all enjoyed this discussion. Thank you for your time. Hope you have a good evening. Talk to you soon.